All right, hopefully everybody sees that. All right, so this talk is hopefully going to be a little, well, it's going to be a little incoherent. Hopefully it makes some amount of sense. I super appreciate that introduction because I am not going to introduce DNA repair pathways at all. Mostly what I'm going to be talking about is non-homologous end joining because it accounts actually for well on 70 to 90% of the repair of a Cas9 generated chromosome break. Typically most targeted double-strand break-inducing nucleases will be repaired by non-homologous end joining. I want to do also almost entirely unpublished data. So very little about what I'm going to talk about is going to have a full resolution. And in many of these stories, you can tell we got a long way to go. They're going to kind of roughly be divided into two parts. The first one really is trying to kind of further advance our sort of long-standing interest in this idea that non-homologous end joining fairly commonly repairs chromosome breaks, including Cas9 chromosome breaks, by incorporating ribonucleotides. So I'm going to talk a little bit about that, but that's going to be a little bit unsatisfying because I'm going to have to stop and then switch gears and talk about what we think is ultimately going to help us a lot with that story, but also has some interesting results in its own right, which is a digital PCR analysis of some early events in genome editing, which talks a little bit about half-life of chromosome breaks, effects of coup on the half-life of chromosome breaks, and then maybe not surprising for some people, but not so surprising for us that there was a significant number of large deletions that are associated with Cas9 generated chromosome breaks and our detection of those. Okay, so first off, this whole question of ribonucleotides on the chromosome arises from our interest in polymerases and non-homologous end joining. Matt mentioned that this pathway is effectively a ligation pathway. You're trying to ligate these ends together, but the really interesting biological sources of chromosome breaks typically don't make a straightforward ligation substrate. So fortunately, non-homologous end joining has this incredibly sophisticated machinery to process these ends. They use a whole bunch of modifying enzymes and include some nucleases, which of course semi-randomly chew into the ends, but then they also have these really beautiful, very specialized DNA polymerases that synthesize nucleotides at the ends in a way to make these ends somehow stickier and the ligase more able to join the end to ligate and finish the reaction. There are three polymerases that account for the vast majority of synthesis during non-homologous end joining. Pol lambda, which I'm not going to talk about, Pol mu, and TDT, and the last two I am going to talk about. Pol mu is probably the most important of the three. It's involved in at least 30% of damage-induced breaks, we think. In fact, pretty much any damage-induced break that has a 3' overhang and is mostly not complementary. That, as it turns out, means that it is not a great, Cas9-induced chromosome breaks are not a great substrate for Pol mu because they don't have 3' overhangs. But you know, I want to show you a model that is actually relevant to Cas9-induced chromosome breaks, which is the TDT model, which I'll get to in a minute. This enzyme has many of the phenotypes, when you knock it out, as many of the phenotypes that you expect from non-homologous end joining deficiency, including radiosensitivity. In a mouse, there's some hematopoietic failure, premature cellular senescence. Most of these are mild, and maybe non-homologous end joining factors are more mild than the non-homologous end joining factors you're more familiar with, things like COO and LAG4. But they're very much in the same vein, saying it's a significant factor that's involved in non-homologous end joining. There's another polymerase that I'm going to talk about, at least show some data, which is TDT. TDT is going to behave very much like Pol mu in the sense that I'm interested, which is this whole ribonucleotide story. In fact, it's indistinguishable in that sense. But it is typically normally expressed only in lymphocytes, and in fact, only in 1 or 2 cell cycles during lymphocyte development, only when you're doing VDJ recombination. When it's expressed, it engages 70, 80% of intermediates at least. We see certainly evidence of this engagement in terms of non-templated additions in about 70% of products. And when knocked out, it results in immunodeficiency, and the reason for that is it adds these non-templated additions that diversifies the antigen-specific receptors you rely on for your adaptive immune response. All right, so either one of these polymerases, whether it's Pol-mu, which adds complementary nucleotides, or TDT, which is template independent, regardless of the source, both of them make ligation easier or needed to make ligation easier to permit ligation. We had thought initially that was solely due to the fact that in the case of Pol-mu, it adds a complementary nucleotide. In the case of TDT, it eventually adds a complementary nucleotide. But I'm going to mention to you just in passing review relative to something we published. That's only really part of the story. Most of the story turns out to be the fact that the reason why these things get ligated by ligase 4 is because when these polymerases are active, it adds a ribonucleotide. Well over 90% of the time, we think, certainly as early as we can detect it, is over 90%. And its reason why it's adding a ribonucleotide is because it has to. If it adds a deoxynucleotide, that ligation is unable to handle the opposite strand mispairs or gaps that would otherwise block ligation, and so you need it to have the ribonucleotide. We think then, and we think that's only relevant for the first strand, this is now a model, we think then that the other strand is repaired without RNA because it doesn't need to be. This is now a complementary, a continuous strand. It's single strand break repair. It can be repaired by deoxynucleotides, and you're probably safer repairing it with deoxynucleotides. Then you go back and do, I guess you could call it the third strand. In fact, it's the first strand. You don't want ribonucleotides in your genome, so you have to replace this with DNA. You do that with ribonucleotide excision repair. In extrachromosomal substrates, we were able to show all this. And the other thing that was kind of striking about that is that steps 2 and 3 were extremely fast. Half-life of less than 10 minutes, probably more on the order of minutes. So it's very fast and very efficient. So we wanted to look at this in the chromosome. And we did a little bit of this in this paper that I'm just citing up here. I'm going to show you a somewhat more high-resolution version of this that we've done since then. It has a little bit more information. Oh yeah, some other things I should mention that we've done since we published this. We've added a couple cell types. It's not specific to mouse cells, which is what we did in this paper. Pretty much every cell type we've tested, we see this. The other thing that was striking for us is it is independent of the cell cycle. There are vastly more deoxynucleotides in S phase. We had thought that maybe these polymerases, because they could actually add both ribonucleotides and deoxynucleotides, they would add deoxynucleotides in S phase. They do not. They still only add ribonucleotides. So these things are adding ribonucleotides in the course of repair. We want to look at this in the chromosome, right? Most of that other stuff I talked about was with extrachromosomal substrates. We want to look at this in the chromosome, so we used Cas9 as a source of a targeted chromosome break. I mentioned this is a terrible substrate for the most part for Pol-mu. So in order to get more abundant products that might contain ribonucleotides or not, and we could look at the fate of these things, we overexpress TDT. TDT, you can, first of all, it's more efficient at these substrates. It adds nucleotides in roughly 90% of the repair products. You can recognize these fairly easily because they're insertions of 1 to 20 nucleotides. Non-complementary sequence, so it stands out. It's not exactly random. In fact, it favors G, and so you can actually capture a lot of these products by amplifying the family of products that contain insertion of 2 or more Gs. That accounts for 20% of all repair. So a fairly large fraction of repair we can capture using a sensitive allele-specific qPCR. So we did that. And this is the kind of data you get. You can see as soon as you introduce this Cas9 chromosome, this ribonucleotide protein product, you very rapidly accumulate repaired products. That accounts all for all products, and I'll get the distinction between all and not all in a minute. As I mentioned, over 20% of all products have this plus GG, which is a TDT-dependent product. When you then compare this qPCR with one where we've treated it with alkali to destroy ribonucleotide-embedded strands, you destroy over 90% of those products in the first, say, 3 or 4 time points, right? So that's up to about 15 minutes. But you can see, as you go along, you're starting to accumulate you're starting to convert, apparently, these ribonucleotide-containing products into deoxynucleotide-containing products. More or less consistent with that model I showed before, where you pair the first strand with a ribonucleotide, so it's almost all ribonucleotides. You would pair a complementary strand with deoxynucleotides, so it should be 50% ribonucleotides. And then because this is RNase H2 knockout, it should stop there. But because we're now waiting a really long time, we think, in fact, you start to dilute this out with replication. But you know, what this really told us is that it's sort of more or less consistent with this. But for one, right, it doesn't look like it's taking minutes. It's taking much, much, much longer in the chromosome, we think. We don't know this because we're putting in the ribonucleotide protein complex. We don't exactly know what's broken and what's not. I'll mention too that there's some other data comparing RNase H2 complemented versus not complemented that suggests that we may also be incorporating ribonucleotides in both strands, but that's a little open yet. Bottom line, we need a better way to look at repair. We need to be able to look at a lot of things at once. We need to be able to probe the state of both of these strands of the chromosome in cells over time. We need to, in terms of state, we need to ask whether it's broken or repaired. We need to be able to identify the strand. We want to know what it's repaired with. We want to know whether it's repaired with a ribonucleotide, with a deoxynucleotide. We want to know the order of that. If it's repaired with a ribonucleotide and then it's replaced with DNA, when does that happen? How fast does that happen? And then we want to get the sense of whether that's actually coupled to non-strand joining or not. We have big dreams, right? We're a long way from realizing that. But one of the technologies that we decided would help us answer these, and we're, you know, making some progress, I'm hoping you'll be able to agree, is digital PCR. So what is digital PCR? It's a single molecule technique. What you do is you take a conventional PCR reaction, or at least this Bio-Rad's version of it. You take a conventional PCR reaction and you emulsify it. So you wind up with something on the order of 16,000, 18,000 little droplets, where each one of these is an individual PCR reaction. And instead of now comparing, you know, relative amounts of product in the linear range, you amplify these droplets to completion. So it's 40 cycles. And the idea is, if there's a molecule in that droplet, it's going to be positive. If there's not a molecule in that droplet, it's not going to be positive. And so it's a Poisson kind of thing. And it's single molecule, which is the beautiful part about this, right? It is precise. So in the case of, here's Bio-Rad's little figure for this. If sample 1 has no targets in it, you'll get no positive droplets. This is a TACMAN assay, it's fluorescent. If it's got 6 targets, you're going to get 6 positive droplets, and you count every single one of these molecules. Same thing as you get higher concentrations, right? As long as you're below saturating amounts, you can accurately quantify the number of events. And I should have mentioned, the way you quantify these events is you basically take this reaction and you stick it in a microfluidic device, or at least this robot does it for you. You flow it through, and there's a detector that detects whether it's positive or not. In fact, even better, you've got 2 channels, so you can actually look at 2 molecules, sorry, 2 amplicons in the same droplet or molecule of target. Because it's a much less complex reaction, right? In the sense that if you don't have, I don't know, 10 to the 9 base pairs to look through, because it is a much more simple reaction, it is a much, much more reliable PCR than conventional qPCR. It is much more linearly responsive, and I'll show you some evidence for that in a minute. You'll also have, as I mentioned, 2 or more channels, so that means you can independently score copy number for multiple amphicolins in the same target molecule. All right, so all that sounds, I don't know, maybe hopefully that made sense. I think what'll make sense though is an example of the actual data you can generate. So what I'm looking at here is the site of a Cas9 target site. That's going to be our target. We made our amplicon like this, where there's a reference amplicon that's in blue. It's a FAM. We have another primer that's a reverse here, and this is a hex probe. If this is like the germline, if this is the original starting substrate, it should be positive for both. And indeed it is. You can see this kind of 2D plot. It looks like a flow plot. You can see that using FAM on this axis and hex on this axis, droplets are either negative, so it's got neither, or it's got both, as you'd predict from an intact chromosome, or having no chromosome at all in that droplet. Contrast that then to if you break it, the droplets should now be blue, but not green. And indeed they are. You can see 100% broken population, they're all blue, none of them are green. And you can mix these two populations to get, whoops, 50% broken, right? So 50% blue, 50% both colors. Indeed, you can do a standard curve. And I used to do double-strand break assays with Southerns. In fact, we were reasonably good at this. It was messy. My nickname when I was a postdoc was Chernobyl because the lab usually wound up significantly hot when I was done. This, I almost cried when I saw it. It is so much easier. It is certainly means the lab is less radioactive when it's done. You get this beautiful response for chromosome breaks. It's extremely linear. Since the efficiency is about 97% for detection. The lower end sensitivity, we can detect anything over 2% broken. It's just a beautiful assay, right? So this is a standard curve basically where we mix broken chromosomes together with unbroken chromosomes. Wonderful way to look at chromosome breaks. In fact, this data is generated experimentally by taking populations of chromosomes that we've broken with XBA and mixing them with populations of chromosomes that aren't. But you can do the same experiment with Cas9, of course, in real life. And this is what you get, comparing wild type cells or isogenic Q70 knockout cells. And what you can see is that you break chromosomes very fast. This is within 15 minutes. And then it stays, it actually never seems to get much higher than that. And so you might think, well, maybe we only broke 35% of the chromosomes. I'm going to show you other data in a minute. That's definitely not the case. We edit by about 12 or 24 hours, 97% of these alleles that we can amplify. So more on that in a minute. In a Q70 knockout, you can get already suspicious that something else is going on here. In a Q70 knockout, there's roughly twice as many that are broken, telling us that, in fact, even early time points, there must be a fair amount of repairs going on, and we'll get into that. Okay, so we can look at repair. So this is now wild type cells only. That yellow thing is that curve for wild type cells that I showed you a minute ago. And now what I'm adding on top of that is a parallel analysis of indels that we got from TIDE of the same samples. And you can see there's an initial rapid breakage. The breakage kind of stays the same, and eventually you repair most of this stuff after about 12 or 24 hours. This is now getting pretty close to the background of the assay. And then a really rapid accumulation of these small indels that we're used to talking about in terms of end joining pathways. This probably includes both alternate end joining, or at least theta-mediated end joining component of alternate end joining, as well as classically defined non-mixed end joining. Most of these are made within a couple of hours, 4 hours, and then there's maybe a gradual accumulation. I think that's probably the theta-mediated end joining component that's a little bit slower. In fact, let me just go straight to some conclusions that I want to draw from this. Perhaps, maybe a little bit of an overinterpretation, and related to the whole short part of this whole short sorted life and times of a chromosome break. Breaks are made really fast. We think over 70% of the chromosomes are broken within 15 minutes. We think the reason why only half the levels of chromosome breaks are present in wild type cells is because, in fact, almost end joining, relative to the Q knockout, is very fast. It'll repair chromosome breaks within tens of minutes. And that explains why the steady state levels are twofold lower. Worth pointing out, interesting, when you overexpress DDT, the steady state levels of breaks are in fact another twofold lower than that. They're down to 15%. Okay, but it then take wild type cells about 12 hours to really finish this whole thing off. And as to what the explanations for that are, now we get into the overinterpretation zone. One possibility is it could be futile cycles. I think this is a fairly major fraction of this thing that's taking a long time to repair this. What I mean by that is you break it, nullus end joining repairs it perfectly, and that's, once again, a Cas9 substrate, so then it re-breaks this. This kind of thing has been seen with ISC1 by Jeremy and Maria. It's been seen by HO endonuclease way back in yeast by Jim Haber, and I think it's clearly happening with Cas9 to some extent. Some breaks will engage alternate end joining, which is slower. I think I kind of referred to that in the last slide. We think that's actually the slower part that takes more like 6 to 12 hours instead of 4 hours. And that, I think, explains most of this. All right, so that's all I really want to say about these fast, accurate indels for now. Of course, we're interested in doing this in the TDT overexpressor. We're interested in looking at the ribonucleotide containing a portion of that. We should transit to the sorted part. I mentioned that this thing edits to about 95 to 97% efficiency, but this graph I topped out at somewhere around 90%. So what explains this kind of stuff, at least up here, this gap? So that gets us into large deletions. So we probe large deletions by now switching our digital PCR setup where the reference amplicon is now 20 kilobases downstream, and the experimental amplicon is what used to be our reference amplicon. So what that means is that we no longer have much information about whether the locus is broken or repaired, but now we can ask whether the flanking side of this chromosome break is deleted or not relative to this 20 kb amplicon. So when you do that, you can see as much as 8% of the wild type cell has lost that proximal amplicon within 4 hours. It eventually accumulates to a total of 14%, and it's about 3-fold more frequent in Ku knockouts.
Hi, Dale.
Yeah.
I got to jump in here. We're out of time.
Yeah.
We have to move on to the next speaker.
Sorry. Okay. I'm just going to show that, and I'm done.
Yep. Thank you very much.
All right. Thanks for the organizers for giving me the opportunity to present some of the work in my lab aimed at understanding end joining outcomes. And what I'll tell you about today are some hallmarks of canonical non-homologous end joining, and then the contribution of both canonical non-homologous end joining and alternative end joining to mutagenic events, as well as alternative end joining's role in oligonucleotide templated gene editing. And frankly, a theme that will be throughout is I'll be describing a few different assay systems and that we've deposited in Adgene that I think that might be helpful for people if they have their favorite small molecule or gene that they want to see might affect repair outcomes. These are available. So in addition to gene editing, you know, a lot of times in the lab, we're thinking about the understanding chromosome break repair outcomes as in the relevance to cancer biology. So for one, chromosomal rearrangements like large deletions are associated with cancer etiology. So if we understand chromosomal break repair can lead to these sorts of deletions, can give insight into cancer development. A lot of what we think about is it's trying to improve a cancer therapeutic response to clastogens such as radiotherapy, where still about 500,000 patients in the US are treated with radiotherapy as part of the first-line treatment. So sticking with radiotherapy and radiation response, I want to use that to introduce the two main pathways of chromosomal end joining. One is a canonical non-homologous end joining, and that's what Dale largely talked about. That involves DNA ligase 4 and associated cofactors. Alternative end joining was sort of initially defined as to explain the phenomenon that chromosomal end joining still persisted at pretty good levels in the absence of canonical non-homologous end joining. And this involves a chromosomal break end resection to reveal single-stranded tails, and then Pol-Q or DNA polymerase theta to bridge ends with microhomology and fill in the gaps. So work from the Tidesterman lab and also Dale's lab has published similar results indicates a marked synergistic role of these two pathways in radiation response. So certainly the absence of the canonical non-homologous end joining, what we're looking at here is a radiation response curve. With loss of CNHEJ causes a marked radiosensitivity. A loss of Pol theta causes a modest radiosensitivity. But the double mutant is markedly dead, even at a relatively low dose of 2 grays of radiation, which is therapeutically relevant because this is a common dose fraction. So if we could try to get sort of this amount of cell killing from a tumor with 2 gray without damaging the margin, that's one of the goals of sort of radiosensitization. Similarly, we're interested in understanding these pathways. As I mentioned, understand cancer etiology. I'll be describing a little bit about deletion rearrangements. Here's just some relevance of that, across lots of cancers, you can see large deletions that lead to loss of key tumor suppressor genes, where the average deletion size across many cancers is about 0.5 megabase. But then this landmark study from earlier this year looked at a wide array of rearrangement types and the amount of microhomology involved. Because as I mentioned, alternative end joining, which is also referred to as microhomology mediated end joining, uses microhomology to bridge the gap across the break. And certainly there are rearrangements that are consistent with canonical non-homologous end joining, in the sense that there's no microhomology. In fact, those are the most frequent. But there's a lot more microhomology and even longer stretches of homology expected by than would be expected by chance. And so sort of understanding the interplay of these factors and how they're regulated can provide insight into how these pathways go wrong and lead to rearrangements during cancer development. So starting with alternative end joining, several years ago, my lab published an assay to look at alternative end joining, which used the rare cutting endonuclease IICE1, where if we have an inactive GFP cassette flanked by 8 nucleotides of microhomology, which if used to bridge the gap across the break, restores a GFP cassette. We compared that to assays that measure IIC1 repair by homologous recombination, and then to provide a contrast with that, develop an assay for total end joining, where a breakage of 2 tandem IIC1 sites and then repair of the distal ends leads to creation of GFP expression cassette. And use this to find that the end resection factor CTIP and other factors are important for alternative end joining and common with homologous recombination, but not required for total end joining. But then, you know, this assay was not specific for either alternative end joining or canonical non-homologous end joining. And part of that is the limitation of IIC1 that creates a 3' overhang that can be repaired by either pathway. So assaying CNGJ proved more difficult with this technology. And so part of that is like, well, what's unique about canonical non-homologous end joining? Well, it's that you know, DNA ligase 4 has a series of cofactors that can promote short range synapsis to allow for LIG4 to ligate these ends without requiring an annealing intermediate, which is not necessarily true about LIG1 and LIG3. So the idea there is if LIG4 might be particularly required to repair blunt-ended breaks not stabilized by an annealing intermediate. So how to test that? Well, we used S-pyogenase Cas9 to create, which largely but not entirely creates blunt-ended double-strand breaks, and then to test whether CNGJ is required for that. So to do that, we needed to create 2 blunt-ended breaks, because you know, say here, if Cas9 makes 1 blunt-ended double-strand break and it's repaired precisely, well, that just restores the chromosome, and that doesn't cause a genetic alteration. So we made 2 tandem Cas9 breaks to excise this 46 nucleotide insert at a region of GFP that's critical for fluorescence. So only if these blunt ends are joined together does it restore this glycine 67 codon and restore GFP positive. So any sort of indel mutation or creation of use of an annealing intermediate to bridge these ends is going to lead to loss of this codon and loss of fluorescence. So each of the assays I'm going to talk about today are in the chromosome. A lot of what I'm going to talk about is in mouse embryonic stem cells, some in human cells, where, you know, like if we look at the frequency of this event, normalized to transfection efficiency, is about half of all cells. So, you know, half of all the cells, if you create 2 tandem breaks, does a precise end joining event with the blunt ends. And that gets to what Dale called the feudal cycles of breakage and religation, that, you know, probably a lot of the time the indels and certainly the homologous recombination events are dependent upon the fact that, you know, canonical non-homologous end joining most often just puts the break back together, but then that recreates the Cas9 cleavage site, creating repeated cycles of breaking and religation. But here, since there's probably a high efficiency excision of this insert, there is no futile cycle for canonical non-homologous end joining, a precise event that is probably the most frequent event that we've seen. Now, this event is absolutely dependent upon canonical non-homologous end joining in the absence of ligase 4 cofactor XRCC4. It's wiped out, but then we can complement that. Same thing for the XRCC4 like factor and Q70. So, so part of what this can be used for is to evaluate other factors that are implicated in non-homologous end joining. Are they important for this no-Wendell event or not? And so a paper published earlier this month from Dan Durocher's lab looked at clustering of factors based upon DNA damage sensitivity, and found this factor ERCC6L2 clustered with LIG4 and XRCC4 and XLF, and then I used the no-Wendell end joining assay developed in my lab and found that cells deficient in ERCC6L2 are important for this particular hallmark of non-homologous end joining. Then also in this cluster of factors like 53BP1, which has been kind of largely miscategorized, I think, as a non-homologous end joining factor. It clearly is an anti-homologous recombination factor. So if you look in BRCA1 deficient cells, homologous recombination is deficient, but then a loss of 53BP1 causes a marked restoration of this event. But then if we look at sort of high fidelity end joining in a 53BP1 null cell, it's no different than wild type cells. So this hallmark of NHEJ can help distinguish between factors that are kind of part of the core CNHEJ complex versus ones that affect repair via distinct mechanisms like 53. So, you know, we haven't solved the 53BP1 question, but, you know, like what are the end joining events that do require 53BP1? But we've also developed this assay system to get an unbiased look at different end joining repair outcomes. I just want to raise this because it's really adaptable to sort of anybody's human cell system. So we use the CD4 gene, which is largely only expressed in T cells, as sort of a reporterless reporter assay, in the sense we use the endogenous CD4 gene that's not expressed in the vast majority of cells we're growing in the lab. And it turns out to be about 1/4 of a megabase downstream from the GAPDH gene, which is expressed in all the cells that we're growing in the lab. So if we create 2 tandem double-strand breaks, a deletion rearrangement will put GAPDH next to the CD4 gene, and then you can just stain the cells with antibodies that bind CD4, since it's a cell surface receptor, and get both the frequency of the rearrangement, and then sort the CD4 positive cells. And then we've done deep sequencing to look at deletion rearrangement junctions. And what we see here is in the first set of bars is what I've talked about so far, that in you know, in parental cells we see a good amount of this no-Wendell junction, meaning the 2 edges of the Cas9 breaks are repaired without insertion deletion mutations. And then in the absence of XLF, which is a CNHJ factor, these events are wiped out with a concomitant increase in deletion mutations. But then there's this other class of insertion mutations that are also dependent upon XLF. So to follow that up, you know, a common insertion event that a lot of you will see in your deep sequencing, particularly of a single Cas9 break, is a +1 insertion. And a likely etiology or mechanism of this relates to sort of the lack of high specificity for cleavage of the PAM side. So the predominant Cas9 break is a blunt end, but sometimes, and it appears to be sequence specific, it'll cut 1 or 2 bases upstream, leading to a 5' overhang. So we wanted to see if we could use this system to look at events leading to a +1 addition. And in this case, we use the same reporter assay, where again we need to fuse the GG and the C nucleotide to create GFP positive cells. But now this Cas9 break cuts between the 2 Gs, where then repair of those 2 blunt ends leads to a frame shift. But if on the PAM side it creates a 5' overhang, and then that's filled in, then that would restore GFP positive cells. And then and in this case, which you know, a lot of you will see in your deep sequencing of Cas9 breaks is +1. we find that that's also a hallmark of CNHJ and also sort of absolutely dependent upon XLF. And that makes sense because, fill in of this 5' overhang is going to just create 2 blunt ends, and then repair of those without a use of an annealing intermediate is going to be dependent on the LAG4 pathway. So what I've told you about so far are the no-indel and +1 insertion events that are dependent upon canonical non-homologous end joining. But what about mutagenic end joining events? So when does CNHJ start to be less required for these events, and then the alternative end joining starts to become important? And for that, we developed, used this platform and added increasing amounts of microhomology. And the upshot of this is that once the microhomology hits about 4 nucleotides, a canonical non-homologous end joining starts to become dispensable, and then Pol theta starts to become important. But I want to dig into this a little bit because, I mentioned before that, in the absence of CNHJ and Pol theta, it's not just an additive radiosensitivity, it's a synergistic radiosensitivity in the absence of these pathways. Sort of indicating that, while a canonical non-homologous end joining might be dispensable once a microhomology is used and Pol theta can start to take over, in the absence of Pol theta, probably CNHJ is important for these kinds of events. And so I just want to provide evidence for that. So, for that, we created a Q70 Pol Q, a double mutant line, and then looked at this no-indel event or events using 4 nucleotides of microhomology that are either at the edge of the break or embedded a little bit. And as I described before, if we look at the no-indel end joining event, only Q can promote that. Adding Pol Q back doesn't work. But now, if we get rid of Pol Q, and Q, these microhomology events are also wiped out, but can be promoted by Q70. So, if Pol Q is, or polymerase theta, and then if the microhomology is embedded from the edge, Pol theta is much more proficient at promoting these events compared to Q70. And I just raised that because that's, while we think CNHJ, while it's known that CNHJ is not required for events, mutagenic end joining events, it does play a role, particularly in cells deficient in Pol theta. And then just in the last data slide, want to talk about briefly another study we published looking at the role of Pol theta in homology-mediated events, which I know a lot of you are working on. In this case, we're looking at oligonucleotide templated events templated by just microhomology. So here we just take that same reporter I've been talking about a lot, but now we deleted 7 nucleotides, and so that any sort of end joining event is not going to be able to restore GFP positive unless we provide it as a donor template. So to look at microhomology mediated events that involve short synthesis, we created these short donors that have a flanking microhomology and then have to fill in the 7 nucleotides that are missing. And then, the notation here is, 12 means 12 nucleotides of homology on one side and on the other, and 7 nucleotides to fill in. It becomes more proficient with 20 nucleotides flanking, but is pretty proficient with 16 and 18. But along the way, each of these events are promoted by Pol theta. So, with these short amounts of microhomology, these are likely not mediated by homologous recombination, but rather alternative end joining, and probably there's a contribution of some of the polymerases that Dale was discussing before. And so we think that this assay system can provide a sort of a more sensitive way of looking at the role of Pol theta in chromosomal break repairs, because it involves not only microhomology annealing, but also synthesis. So that's the last data slide I wanted to talk about today. I can go back to the summary if people have, but I just want to leave time for questions. So here are the people who did the work. Most of the end joining work I described was done by Ragini Bhargava, a former PhD student who's a postdoc in Pittsburgh now. And I talked about a little bit about Jill Tye's work, and you're going to hear about the rest of that in a little later. supported by a research associate, Wednesday Lopez, Colorado, and then the last oligonucleotide templated experiment was done by postdoc Drew Kelso. Thank you all for your time, and I'll be glad to take questions.
All right, thanks.